Extracellular vesicles and exosomes are transforming how we understand disease, diagnostics, and delivery. At Atlantis Bioscience, we offer a comprehensive exosome workflow solution, supporting your path from bench to bedside. To begin, your EV research starts with healthy, well-conditioned cells. Our cell culture solutions include BioWest EV depleted FBS, exosome depleted human platelet lysate, and extract media that are chemically defined for clean exosome harvests. Once your conditioned media is ready, the next task is reducing sample volume efficiently. With the QEVTFF, you can gently concentrate your samples while maintaining EV integrity. You can achieve high-purity isolation using QEV-SEC, either manually or in combination with the ISON AFC. For higher throughput needs, consider using QEV-Dexter. And if you're scaling up, QEV-Zenko offers a robust solution for large volume EV isolation. After isolation and concentration, it's essential to validate your results. The Exoid system provides real-time measurement of size, concentration, and zeta potential for nanoparticles. And with the ExoBright series from Biotium, you can explore total RNA content, perform Western blots, conduct flow cytometry, and label EVs with true EV lipophilic membrane dyes optimized for high-specificity detection of extracellular vesicles. At Atlantis Bioscience, we work with researchers across scientific, clinical, and veterinary fields. Our portfolio includes over 1,000 products, from cell culture media and viral vectors to advanced reagents and analytical tools. Find everything you need at Atlantis Bioscience. Visit www.atlantisbioscience.com. Atlantis Bioscience, your lifetime partner in translational science.